Yeah. Yeah.
No, I was like, I'm like, hopefully somebody else show up, or he gonna be like, well, nobody showed up today.
Quiz time. No. I would just lecture to myself. Okay. Anyway, so we're going to pick up where we left off because, unfortunately, we got a lot to cover and we got about 4 weeks left. So, yeah. So now, if you can make up a similar little tune for, I don't know, to help out with this, yeah. It's like the Megalito pipetting song that helped me keep track of what well I was in. It's just something silly and dumb. Anyway, where we want to pick up with is hormonal signaling, and we're going to first talk about top-down versus bottom-up signaling. So we really think top as the brain, as the head, that's our control center. So top-down signaling is going to be hormonal control from the brain, and bottom-up is going to be hormonal control from the other tissues that send signals to the brain. All right. And we'll look at the first examples of top-down as oxytocin, vasopressin, and cortisol, and then we'll talk about epinephrine, insulin, and leptin. And you guys are going to get to see the best picture of the entire semester here coming up in a little bit. That's your incentive to stay awake. Okay. But the hypothalamus is our central coordinating system. And if you think about some of what Disney Pixar movies, what is it, Inside Out, or I can't remember, but then they do a passable job of there's enough stuff in there to make it it's like, okay, this is okay. But our hypothalamus is that small region in the forebrain of animals. It's going to receive and integrate nerve signals from every place else, and it synthesizes peptides, oxytocin, and vasopressin. So these are very small molecules, and these regulate the function of the anterior pituitary. So we'll see a picture of where that is. So the posterior pituitary has the ends of axons from the hypothalamus, and the anterior pituitary, which is in front of that, receives the releasing factors from the hypothalamus from the blood vessels, and it produces various tropins, and it activates multiple targets in there, like the adrenal cortex or the thyroid or the ovaries or testes. So comes from the hypothalamus, goes to the pituitary sends a signal, passes on there. If you are, not really obsessed, but just paying attention to military stuff, it's part of our command control chain in there. Okay. So we can see our hypothalamus sitting up here and our pituitary, posterior and anterior, that are connected in various ways. Okay. I will say one thing I was interested in coming out of grad school, in neurobiology, neurology, all sorts of stuff like that, neuroscience. And so I was like, oh, I'll try and learn a little bit of neural anatomy. Yeah. Good God, everything looks the same. So that's challenging. It takes a lot of self-training to discern the different portions. It's like, well, why is this anterior? All right. It's just a gray field. All right. Well, let's talk about these hormones, oxytocin, vasopressin. Oxytocin I refer to as new baby smell because, that is, when the uterus is contracting, it gets released in there. It also stimulates milk production from the mammary glands. And there's some evidence that it actually functions as a pheromone, and we can see that it plays a role in social behavior such as pair bonding. So why do mothers love their babies? Because the babies are inducing oxytocin, which is the love hormone, something like that. Right? You can see that in other pair bonding as well too. Vasopressin is more of a blood pressure regulatory molecule. It regulates constriction of blood vessels and also absorption of water in the kidneys to maintain a salt balance, so that you don't have a too concentrated you want isotonic solutions, not hypo or hypertonic. But it also has that pair bonding stuff. Here's what they look like, and you can see these are very short peptides. We're talking about what, 1, 2, 3, 4, 5, 6, 7, 8, 9, roughly 9 amino acids, and we see a cysteine bond in there, so we get this nice little kind of ring structure in there as well. Yeah. So, and the only differences between oxytocin and vasopressin are an isoleucine converted to a phenylalanine. Okay, that's pretty big, and a leucine to an arginine. Okay, that is a very there's two different amino acids in there, but they're very big differences. We go from a, medium-sized hydrophobic to this big ring structure, and we go from, a hydrophobic to a positively charged one in there. So those two changes allow them to have, massively different binding affinities and targets in there. So, all right, but you can see that they're probably similar to make. We should also talk about cortisol. If you ever have, injury or you get afraid of something or stress, cortisol is our stress hormone. And so anytime there is something stressful that happens, let's put this down here. Maybe right in the, okay, right there. But anytime there's some type of stress, whether it's injury or, terror or even like hypoglycemia, you get low blood sugar, or if you're in pain, all this is going to trigger, activation of the hypothalamus, which is going to release this corticotropin releasing hormone, which will go to the pituitary, which will release corticotropin, which will go to the adrenal glands, which will release the cortisol. So court, so it's a whole bunch of, it's like dealing with the legal system. There's a lot of time spent in court. Okay, I don't know. Whatever. But the cortisol gets released from the adrenal gland and then affects other tissues. So our signal comes from the brain, and this is the top down, releases the cortisol ultimately to stimulate effects on the muscle, the liver, and the adipose in there. Okay, we're looking at nanogram levels of corticotropin releasing hormone in here. So this is minor. But when that gets to the pituitary, we see microgram levels of corticotropin, right? And then we see milligram levels of cortisol that get released. So signal cascade, we only need a very, very small amount of our initial one, and we get this massive release of cortisol. And of course, cortisol can feed back and inhibit this. Once you have the cortisol that is, dealing with whatever environmental factors are influencing the initial release, it can feed back and prevent it. Of course, if you're constantly in fear, constantly in stress, constantly in pain, then you end up with some other effects in there. Okay, and we'll talk about that in a second, because my slides are, I just looked at these and they're in slightly different order. But let's go back and talk a little bit about bottom up now. On the other hand, our other organs can also function as sensors of various things as well too. For example, when we eat a meal, our intestine is going to release GLP-1 and GIP, which is going to feed back into the pancreas and say, hey, release insulin, or hey, release glucagon, depending on what's going on. Actually release glucagon if we're hungry, but we'll go back here and we'll inhibit the release of glucagon and we'll promote the release of insulin. For the stomach, I always like this. If you get hungry, what does your stomach do? It growls, right? So if you're hungry, you're going to release ghrelin. My stomach is growling. I'm hungry. I don't know. I can't really. What accent is that? Weird one. And it'll go to the hypothalamus and stimulate some activity in there. Same way we can see fat tissue releases a hormone called adiponectin and also leptin, both of which can go feedback into the hypothalamus and have effects in there.
It's funny because when I'm hungry, I like do something like chew gum or something. And then I take the whole just for
yes.
like, I just put a little taste on my tongue, something.
Yes, there is a satiety index as well too. So sometimes even like drinking water, you add volume to your stomach, and that reduces your hunger. And not because you were adding fuel, but you just kind of feel full. Yeah, I know. I'm like,
hey, no, I was sleeping there last night. That's not an option.
But the key here is we have our other organs that are releasing factors that go and affect the hypothalamus. Okay. And I think I mentioned before that, I used to think the brain was the most important organ, but it's really probably the liver, because the liver is the one that's going to adapt to all these changing metabolic conditions, which is what we're going to talk about a lot in this chapter. Okay. So portal vein carries nutrients into the liver. So I'm going to eat my my healthy breakfast of whole grain toast and beef jerky. It's like pure protein. But the portal vein will carry all that into the liver, and then the hepatocytes will turn those nutrients into fuel for the other organs in there. And of course, our hepatic enzymes have rapid turnover. We're constantly degrading them and making new ones. And the enzyme concentrations will increase or decrease as we change our diet, and as our other tissues have certain requirements. Okay. So a lot of this chapter we've seen before, so it's a little bit of a review of this semester. which makes it seem like this chapter is massive, but it's just kind of taking what we've seen before, and now we're going to start applying it to the whole organism. All right. So first, we're going to talk about carbohydrate sensing and processing in the liver. We know that we have glucose or fructose or galactose or whatever our sugars get converted into glucose 6-phosphate. Okay. So we can think of, if you are familiar with any cities where you have like train stations, I always think of going to New York City and Grand Central Station, woo, yay. Or actually, I'm more familiar with DC, but there's our transfer station. The liver is the transfer station for glucose 6-phosphate. We can make glucose 6-phosphate lots of different ways, and we can use it lots of different ways. So if we think of our hepatocytes, they have this glucose 2 transporter, and it allows glucose to come into the liver and go out of the liver. So if we have lots of glucose in the bloodstream, we have our higher concentration in the bloodstream, we're going to see more of it travel into the liver. On the other hand, if we have low blood glucose in the bloodstream, that would be redundant, then higher concentrations in the liver will let that glucose go out into the bloodstream, and it can be carried to the organs that need it. The other thing is, we know that the liver has hexokinase 4, which is going to phosphorylate glucose to make G6P. Once it's phosphorylated, it's trapped in the liver. Who's the guy who's saying trapped in the closet? There's something. Was it R. Kelly?
Confessions.
Yeah, maybe. I think so. Yeah.
No, that was R. Kelly. It was R. Kelly. Okay, I said one of them, but I think Usher wrote it.
I just think of the South Park where Tom Cruise is trapped in the closet. But the glucokinase is going to keep the glucose in the liver. So we have to dephosphorylate to get it out. So free glucose diffuses in or out, but once it's phosphorylated, we know it kind of stays in there, and then we can feed it into multiple pathways. One of the interesting things is our hexokinase 4 is not inhibited by G6P. So there's not a feedback inhibition in there. So basically, if there's glucose, it's going to phosphorylate it. And we can constantly, go away. We're constantly making G6P. Whether we have high glucose concentrations or low glucose concentrations. It just depends on, if we have lower on the outside, some of that glucose is going to diffuse back out. Okay. We already know what can happen to G6P. We can get free glucose to send to other tissues. We can convert it into glycogen to store for medium-term energy. We can go into glycolysis to make ATP for the hepatocytes themselves. We can make acetyl-CoA to be made into fatty acids and triacylglycerols to get exported to other tissues. And it can go into the pentose phosphate pathway to give us NADPH and make ribose 5-phosphate for nucleotide production. All right. So we know all that. All right. And this is just kind of a brief review of that, of here's what's going on in the liver. Okay. And the key is glucose into the blood or glucose out of the blood, or we can, glycogen, triacylglycerols, energy, so on and so forth. Okay. Ribose 5-phosphate. Yeah, we got everything in there. Just a nice little picture of that. Little review, little quiz for Duck. Okay. Similarly, we can look at our amino acids. Many of them are glucogenic. If you read like labels on food, you can see like, oh, this is so we'll see a lot of ones that can be converted into glucose as well in there. We can also, of course, see that amino acids can be recycled or made into hormones or nucleotides, or go into various pathways like the citric acid cycle or gluconeogenesis or whatnot. So we have different fates of amino acids all in the liver as well too. And of course, we can send some of these. You'll see some of them go into the bloodstream and help transport stuff. We're going to look at the Cori cycle up here in a little bit. We have like the alanine coming from amino acids and muscle in there. If we look at fatty acids in the liver, the CAC, citric acid cycle. Yep.
Okay.
It's particularly ridiculous for me because the art center at WVU where I went to school was known as the CAC, Creative Arts Center. So I'm like, yeah. All right. Fatty acids. We can also make liver lipids. We can oxidize them for fuel in the liver. We can convert them into ketone bodies, especially if we have excess acetyl-CoA, and we can send those ketone bodies to keep the brain functioning and the heart and other stuff as well too. And we can also make cholesterol. We can convert them into phospholipids. We can convert them into triacylglycerols and send them to adipocytes for storage. And we can carry them to the heart and muscle for direct fuel there as well. Okay, so internal fuel, external fuel, storage, and things that we need. So we've kind of seen all of this. we're just kind of trying to put it all together here. So the liver processes carbohydrates, amino acids, and lipids. All right. And there are multiple fates for each one of those. You got to know all of them.
Wait, what?
Well, you should. You don't need to memorize all of them, but you should be familiar with the pathways. And that's actually going to bring me to something on the exam. On the very last question, the take-home portion, about the isovaleric anemia, isovaleric something like that. One of the questions I had in there was, what happens if you completely eliminate leucine from the diet? So the thing was, fatty acid, it inhibits propionyl-CoA carboxylase. So, odd chain fatty acids. And most of you, I think everybody said that, it's needed for this propionyl-CoA, but if you eliminate odd chain fatty acids from the diet, you still have the disease. Why? Because propionyl-CoA carboxylase is necessary for the metabolism of 4 particular amino acids: leucine, methionine, threonine. Yep. Glycine can help out because you actually form this isovaleric glycine compound, which reduces the concentration of isovaleric acid and helps reduce the acidemia associated with that.
35, like, you got leucine problem, glycine.
Yeah. But there is one thing that nobody picked up on. Now, of course, it's a small class, but one of the further questions in there was, well, what happens if you completely eliminate leucine from the diet? And it's like, well, that's going to.
I think I said couldn't completely eliminate it because it's found in.
It's found in
just a couple of foods.
Yeah, it's found everywhere. Yeah. But the real reason you can't eliminate it is because we can't synthesize it. It is an essential amino acid, and that's a weird thing in there where you got to make that connection all the way back to essential versus non-essential amino acids. So we have to have some leucine in the diet because we can't make it. Now we can, of course, scavenge, but you're going to see if you don't eat any leucine at all, eventually those leucine, whatever you have in your body, is you can see that concentration plummet, and then you're going to have other issues with that. So damned if you do and damned if you don't. But that's the type of, when I have those take-home questions, pulling this stuff from a lot of different areas. And that's a hard question because, it's not immediately obvious and you don't see that. It's kind of synthesizing that whole big area of knowledge in there. So that's why when I kind of put up these review for what does the liver do for carbohydrates and amino acids and lipids, we're seeing this interconnection. And now we're going to look at how the totality of regulation. So there's a whole lot of information here. And this is what makes Biochem 2 metabolism particularly hard, because there's just so much stuff that affects so much other stuff. Everything is interconnected. We are all one in the universe. Okay. If we look at the acetyl-CoA in there, so we have our lipid pools, acetyl-CoA. If we reduce carbohydrates, we're going to see lots of acetyl-CoA and a lack of oxaloacetate. So we can make ketone bodies in this case, and we see acetone, acetoacetate, and this beta hydroxybutyrate. These can provide up to 70% of the energy needed for the heart during prolonged fasting. These can also cross the blood-brain barrier and provide fuel for the brain. Okay? And we're going to see that the brain requires constant energy in there. Okay, and we'll look at this in a little more detail in a minute. Okay, one thing I find interesting. Fat tissue. We're going to talk a lot about adipose in this, rest of this chapter as well too. We basically have 2 types of fat: white adipose and brown adipose. Our white adipose is what we think of when we jiggle or whatever, Dr. Ebert is slowly shrinking there. That's our big storage droplets, stored fuel for later. And if you eat, lots of crappy food, then you store lots and lots of it. Brown adipose is a little bit different. And we're going to actually see a picture that'll show this. The brown adipose tissue tends to be smaller cells, and they are not, as opposed to having one big giant lipid droplet in there, they have lots of smaller droplets in there. Most importantly, there's a lot more cytochrome in there, which, kind of colors it. Our fat, the adipose by itself is kind of white. That's why it's called that way. And you'll see this picture come up, but there's more cytochromes in brown adipose. There's also an uncoupling protein that allows you to burn the fat and not produce energy, but instead produce heat.
I feel like I should know what a cytochrome is.
So we are looking at those in terms of our electron transport chain. We're going to see lots of mitochondria in there. So our cytochromes are, And so, you got the various cytochromes in there, but they tend to be kind of brownish in color. I don't know. Is it iron, sulfur? Yeah, I'm not sure exactly why, but I can remember watching them separate on there. You see this little brown ring moving down the column for purification. All right. Pictorially, our cartoon, one big fatty acid droplet, a few mitochondria, and our nucleus in there, and that's our typical white adipose. Our brown adipose, on the other hand, you can see we have a lot more mitochondria, so a lot more cytochromes, and our lipid droplets, instead of just one big one, we have a lot of smaller ones, and our cells a little more centrally organized. Now, if you look at this, these are at the same scale. Our white adipose, a lot bigger fat stores, a lot smaller rest of the cell. Our rest of the cell is much more prominent. It's maybe not quite, our cartoon is not quite accurate because we still have a lot of adipose stored in here, but there's a lot more of the other stuff. The big thing is the uncoupling protein that allows this UCP1, sometimes called thermogenin, which allows the proton gradient in the mitochondria to be dissipated without producing ATP. Instead, it releases heat. So thermogenesis. We find lots of brown adipose in newborns. You'll see it in rats as well too, various animals in there. And we find it's localized around important organs. So if we look at our newborn here, and this is not the best picture.
Can we talk about this, like people that live on board?
Yes, yes. Yes, yes. You can actually convert white adipose into brown adipose through cold exposure. So this is our typical newborn, and you can see the brown adipose through the core of the body that helps make sure that the core stays warm. So even when they're cold, as you mature, that brown adipose converts into white. But if you go into cold exposure, you can see, the dark color here. You see brown adipose around the heart, the brain, and the gonads. You spend some time in cold, and that white adipose gets converted into brown there as well too.
People say, oh, they feel cold, they really feel cold.
Yes, yes. And you can do this by, go hang out in the cold room or, whatnot. So you can actually convert that in there. I always think too about, so the functional MRI study group, I was a guinea pig for that, but I found it fascinating. They were actually looking at fMRI signals between Tibetan monks who did a lot of meditation, and then regular people who had no meditation training, and looking at activity centers in there. They actually found with just like a few lessons, a couple weeks of like training, regular people could see this massive spike in the change in there to get a lot closer to that. But one of the stories about the monks is, up in Tibet and cold mountains and snowing outside, and you can take this guy and wrap him in a blanket, in a wet blanket, and like an hour or two later, the blanket would be dry because you just put off so much heat. So theoretically, that's probably a conversion of white adipose to brown adipose, and they're just generating a ton of body heat. And because they live there and don't wear a lot of heavy clothes, so you can get used to it. Yeah.
Because like I always say, like my brother and my dad are hot nature. Like my dad will put on a shirt, and about like an hour later or so, they don't.
Steam iron. Yeah. Yep. Iron it while you wear it. You gotta be tough. Yeah. Yep. So at birth, we all have lots of brown adipose in there because we're going from this nice little 37 degrees Celsius environment out into the cold, cruel world. But as we grow, that declines. But even in adults, we can change this. So if you just hang out in the cold all the time, I joke, if I really want to lose weight, I'm just going to go sit in the cold room for an hour every day and start making more brown adipose. I don't know. Yeah. Or I could just do more exercise. Actually, my leg feels good enough. I can ride the bike again. So I can do that. All right. So what do adipocytes do? I mean, they store fat, right, for energy in here, but there's a lot of processes that are going on within them. We sort of touched on that in the last exam section when we talked about hormone sensitive lipase and cleaving our triacylglycerols into free fatty acids. But adipocytes can convert acetyl-CoA into fatty acids. We can use the fatty acids to make our triacylglycerols. We can get that from intestinal lipids, from the chylomicrons, or from the liver, from the VLDLs. And then when we need energy, they can release fatty acids to the bloodstream to go to the muscle and the heart. The brain, nope, brain can't use fat. Brain needs sugar. Gotta have brain candy in here. Hormones work very quickly on adipocytes. So we're going to talk about epinephrine a little bit. I know we've mentioned this a little bit before as our in terms of second messenger, and it's a GPCR receptor and produces cyclic AMP as a second messenger. But epinephrine gives a very fast response throughout the body. In terms of adipocytes, it will stimulate access of hormone-sensitive lipase to the lipid droplets. So the hormone-sensitive lipase is kind of sequestered away. The epinephrine binds to the receptors on the adipocyte, and those go to the fat cell, hydrolyze the triacylglycerols, and release fatty acids because we need energy now. It's like an even more strident J.G. Wentworth. It's my energy, and I need it now. Yeah, see, all right. Yeah, I was gonna say, yeah, I know. I've got maybe another year or two of those because I haven't seen too many of those, but I don't watch that much TV anymore.
You got Everest College. Nah, the ways they sit on the couch with one teacher using Michael Jackson. Yeah, I keep pressing over and over. Maybe I go to college. Do it now.
If we look at muscles, we've got our two types of muscle fibers in there: slow twitch and fast twitch. Red muscle versus white muscle. Slow twitch is, and it seems counteractive to me the way they're named. I've struggled with this naming forever, but slow twitch has lots of blood vessels and a lot of mitochondria. But the reason it's called slow twitch is because it provides energy through slow and steady oxidative phosphorylation. It's kind of constantly going. It's this background, you're ready in there. The fast twitch, on the other hand, has fewer mitochondria and a lower capability to deliver O2 to it, but it uses ATP faster and fatigues faster due to greater demands, more tension, more less in there. So endurance training can increase your fast twitch in there. You get in there and run on the treadmill for hours and hours and hours. Yeah. Yep. So this is our just basic doing stuff in here. This is me doing yard work, just slow and steady. And then this is me not running away from trees. Yeah, damn it. Where do we get the energy for our muscle contraction? Okay. Typically we're going to see, fatty acids, blood glucose, and all that is going to be, most of our stuff in there from aerobic oxidation. Phosphocreatine can be used in the muscle for bursts of heavy activity, and also muscle glycogen. So we tend to talk mainly about liver glycogen because that's where we store most of our energy. And then when we need it for the short term, the liver will release it as glucose, and that'll go to the muscles for energy in there. But muscles have glycogen too. One of the big differences, though, is muscles don't want to use that glycogen. When you use muscle glycogen, even as you're using it, you're going to start trying to regenerate it. So it builds back up very quickly. But if you need it for immediate access, very heavy activity, very strenuous stuff, you're going to be burning muscle glycogen. Both the glucose and the muscle glycogen releasing glucose within the myocytes are going to produce lactate. So you get that lactic acid buildup, feel the burn, and your muscles feel like they're burning. Literally, that's lactic acid, the acidemia in there. And we need to send that lactic acid to the liver, where it can be metabolized. Okay. Muscle glycogen, much like liver glycogen, you can release glucose 6-phosphates, and you get 3 ATPs out of it and not 2 because you skip this hexokinase reaction in there. So you get more bang for your buck for the muscle glycogen. The same thing with phosphocreatine. Recall that is a higher energy molecule than ATP. So, you can, generate ATP through phosphocreatine. And this is what it kind of looks like here, all the good stuff in there. So that you can make this energy when you need to do something, lift a car off a child or run from a bear or whatnot in there. Mm-hmm. All right. We also see that phosphocreatine plays another role in the muscles. It buffers the ATP concentration during exercise. So here we are with, we're at rest. We're going to have fairly high levels of phosphocreatine. As we go through exercise, you can see our time frame is going back into the plane of the projector up there. Our ATP levels are going to stay roughly constant. But you see our phosphocreatine, we have this massive dip. And of course, this release of inorganic phosphate. And then during recovery time, we're going to regenerate that phosphocreatine. But even during exercise, we're trying to maintain that ATP. Now, if you exercise to exhaustion, then you would see these levels dip as well too. But typically it's just going to be hard exercise and then go back to recovery and rest and build it back up. Okay. Yeah. There's lots of good things out there. The hardest part about these plans is sticking to them. Consistency. All right. So. Yeah, we've seen this picture before. This is just epinephrine releasing glycogen, mobilizing glycogen to release blood glucose. So one molecule of epinephrine, typically in the liver, can stimulate the release of 100,000 molecules of blood glucose just through the signal cascade. Okay, this is breaking down glycogen in the liver. It can do this in the muscle, but you're not gonna. The muscles don't like they're like me, they're tight. They want to hold on to it. It's like, no, I don't want to use this. It's like this PayPal account that I have that I've never taken any money out of. It's only like a couple $100, but it's there. And it's just this, oh, I keep forgetting that I have this extra money in case I need it. And there's never yet been an incident where I've needed that money right away. I've had other stuff that I could use, but that's the stuff that is socked away. Okay, so that's what our muscles are kind of trying to do with their glycogen. If you do really vigorous exercise, you'll probably notice you breathe heavily, and that's because there's an O2 debt. We're using that oxygen for oxidative phosphorylation to build up that proton gradient, replenish the ATP. So we need to get more oxygen there. So you're going to breathe heavy to bring in more oxygen. And you can see, like some sports players, they especially if they're at high altitude, they'll put the little O2 mask on them and give them pure oxygen in there. The other thing we need is we need ATP for gluconeogenesis to take up the lactic acid and restore the muscle glycogen concentrations in there. So this is where we get to the Cori cycle. So here we are in the muscle. We burn muscle glycogen, and we produce our glucose. We get our ATP, so our muscles can contract. But one of our byproducts is lactic acid. So we can send that into the bloodstream. So we see lactic acid in the bloodstream that goes to the liver, where lactic acid gets converted to glucose, requires ATP in the liver. Right? We can make the glucose, release that back to the bloodstream, and go back to the muscle and replenish the glycogen stores that are in there.
So if you have a liver issue, your muscle recovery would be
potentially. Maybe not always. It would depend on the particular liver issue, but a healthy liver is critical to the liver is probably the most important organ because it's just centrally involved in the metabolism of everything else. The brain signals a lot of stuff, but the liver is just grand central station. Everything's got to go through the liver. Now, and this is one of the things that it always took me a long time to kind of understand. Like hockey players will play like 45-second shifts. I'm like, 45 seconds, that's not long, but they're burst of energy to skate and skate fast and do all this stuff, and then they need rest, and then it's just a couple minutes on the bench allows that lactic acid to get to the liver, get converted back into glucose, and go back to the muscle. And so you get rid of the lactic acid and replenish the gradients within the muscle. You contrast that with soccer players. Soccer players are incredibly fit because they're running around the field all the time. But most of the time, they're just kind of jogging back and forth, or there are short bursts of speed there as well too. And then you might see them lag or rest a little bit, so they recover on the field. Whereas hockey players, are pretty much most of the time burst of energy. And it's funny to see like a long shift where they're tired and they can't move. And I'm like, these guys are in really good condition, but they're using so much energy in such a short period of time. That's true. Yeah.
It's like your legs give out, you're gonna fall.
Yeah.
And then my fear is somebody slicing off my head.
It's I've seen, and it's incredibly rare. But I remember I was watching a game where somebody got a skate to the neck And survive. I mean, this was the NHL, so the on ice medical care was phenomenal. Of course, they had a towel on there and applying pressure right away, and I mean, it was immediate surgery, but oof, yeah. Okay, so let's contrast heart muscle versus skeletal muscle. The heart muscle has more mitochondria. It's about half the cell. So yay mitochondria, because that heart muscle has to constantly keep working. Unlike, if I can run around, I can sit down and rest, but my heart has to keep beating. The heart gets fueled by a lot of fatty acids in there. You can use some ketones, some glucose, some phosphocreatine, but fatty acids are its primary source of fuel. So you don't want a fatty heart, but you got to have fat in your heart. It's an aerobic organ. If you cut off the oxygen supply, then the muscle will die. And this is what we see in our heart attacks, our myocardial infarctions, or MI, infarct, whatever terminology you want to use there. Yep. Yep. So if we look at heart muscle in here, you can see the contractile apparatus and the mitochondria are right beside it. Angina, what's the other term there? Yeah. There are various forms of heart disease, but anything that cuts off the oxygen supply is going to be the infarction.
This one they get the nitroglycerin, the tablets if they feel like they have heart attack.
Yes, yeah. Okay, the brain is also very aerobic. It will consume about 20% of the oxygen that you breathe at rest. All right, so if you think hard, you're using up more oxygen, man. So we got all these neurons and glial cells. We use about 130 grams of glucose a day. So let's put that in American. So that's about 0.13 kilograms. So we're looking at, not quite half a pound, probably like 6 ounces of pure sugar daily. Yeah. It's very dependent on glucose. It can use beta hydroxybutyrate, those ketone bodies, and the astrocytes can use fatty acids, but the neurons need sugar. You gotta have brain candy. It's also important that we have this ATP to maintain the potential across the membranes of the neurons. So if you think of the electrical activity in there, if we don't maintain the electric potential, then we're not going to get neurons firing, and that's bad. Okay. So during the normal diet, glucose will go to the brain. We'll use tons of glucose for the brain. During a starved state, we'll use ketone bodies. Okay. Both of those can feed into aerobic oxidation and produce CO2. And the key is we need that ATP in there to maintain our electric potential. Okay. I don't know if you guys donate plasma, extra money, the beer money. That was like the big thing when I was an undergrad. They literally there was, yeah. I knew so many people who'd go to Saratech. Yep, there's a plasma donation center that was literally across the street and like one building down. There's like the second building off campus was the plasma center, and having gone to a school that had a reputation for partying, I knew a lot of people who went and sold plasma for beer money.
I think if you have like old blood, I think it's great for you though.
Yeah.
You're helping so many people. But then like mine, they were like, girl, we don't need yours. Like there's some people where I would help, but
I'm pretty sure I'm AB positive. So
I have no idea. Yeah. We never did the experiment.
Yeah, I think that was from blood typing back in high school biology. Yeah. No. And in my case, it's probably mirrors me because I must collect everything. I must have everything. So I got the A isoform, I got the B isoform because each one of those have an extra little carbohydrate group on there. And I'm positive, so I got to have the rho factor in there too. So yeah. Although I'd figure I'd be negative because I'm so.
I thought they put it on like I said I thought that too. I know it's either one or either like whatever your parents are. My dad said he like he don't even. I'm like I'm gonna be on emergency. Hopefully I won't get worried.
Yeah, I was gonna say, and if you ever in that case, I mean, they can type it so fast. That's probably why it's not as because I don't remember. It's a quick test. Right. But interesting thing about blood, our erythrocytes, our blood cells, lack mitochondria, so they rely purely on glycolysis. We have to maintain them in an isotonic solution. That's why if you actually ever get an IV, it is probably going to be 0.9% saline because that's the same concentrate, the isotonic concentration to maintain that. For feeding, you would see a 5% glucose solution because that gives you glucose in the bloodstream, but it's also at an isotonic concentration in there. We can see, we're made up of the cells. We got erythrocytes, leukocytes, platelets in there. The plasma itself has various plasma solutes. You look at all these organic metabolites and waste products that are in there. There's a lot of urea in there, and so on and so forth. All sorts of other little plasma proteins that play different roles. Glucose levels, important. So you have to maintain them in a normal range. Too high is bad, too low also gets bad. So if you don't eat for a while, you'll start seeing subtle neurological signs of low blood glucose. Like you get hungry, your body will start releasing glucagon or cortisol as necessary. You get down to about 50, you might sweat or tremble, you get like little shakes a little bit. It's like, oh, okay, I need to eat something. I haven't eaten all day. You get the shake there. When you start getting below 40 is when it starts getting very dangerous, and that hypoglycemia sets in. You get very lethargic. You can't move.
So that's like fasting. And some people can get that low.
Under normal circumstances with fasting, your body is still going to be releasing. So this would have to be in late stage starvation. Usually, your body is going to work to maintain glucose. And we're going to see a slide here, one or two slides coming up, that's going to show how glucose levels go over the course of like a month of starvation. So your body is going to work to prevent that. But, there are disease states where you become hypoglycemic, like diabetes. That's where you get to diabetic coma is where your blood sugar drops so low here. And if your blood sugar gets down to like 10 mgs per 100 mils, 10% in there, then we're looking at permanent brain damage and death as stuff in there too. Right. So ideally, we're in this range, 60 to 90. That's why if you ever do like the blood test for blood sugar, you fast overnight and they go in and draw your blood, it should be between that range. If you eat, you're going to see this being a little bit higher because you're going to have more glucose in there. I think of that because I went like right after I ate a snack, I ended up going someplace and having a blood draw, and they were reporting it's just a general panel of stuff. I was like, oh, my blood glucose is 99. I was like, oh, well, I did just eat right before that, so that's probably normal because I just had a bunch of food. Yeah. So, but fasting, it's normally in the 80s, I would think. Insulin is going to stimulate conversion of excess glucose to glycogen or triacylglycerols. We kind of know all this. So our insulin is important in glucose uptake in muscle and fat cells. Okay. In the liver, it's going to stimulate that glycogen synthase. Also in the liver, it's going to stimulate fatty acid synthesis in there. So different organs are going to respond to the same hormone in different ways. That's what we're looking at here. Okay. And all this chart does is like, hey, look at all the things we've learned in the past. All right. So this is where it all plays. We're looking at carbohydrate catabolism, carbohydrate anabolism. Where does this happen? In the same way, we can see this illustrated in chapters here, all across the stuff that we've talked about. These are just kind of, if this sounds familiar but you don't remember it, this is go back and check out this figure. Go out and check out this figure, and it'll show you what you've got. Yes, yeah, and it's probably not enough. Well, not terrible. Yeah, because there's so much information. But what we want to get across though are the basics in here. So, what time frame? Okay, good. I want to talk about the well-fed liver versus the starved liver. So you wake up and you have like bacon and eggs and cereal and milk or something like that. You got lots of food coming in there. You're going to see your intestine. You got amino acids, fats. What was it? Big glass of milk here. Yeah, amino acids, carbohydrates, and fats all in milk in here. Our triacylglycerols can go into the lymphatic system and go directly to the muscle. They can also go to the liver, and where they can accumulate in VLDLs and get sent to the adipocytes for storage. All right. Our amino acids and our glucose go into the blood vessels and through the portal vein, get to the liver. Our glucose we can convert into glycogen. We can send it through glycolysis. We can go into the the pentose phosphate pathway, and of course we can send it from the liver to other tissues like the brain to use as energy. Our amino acids, same thing in here. We can have our ketogenic and glucogenic amino acids in here. We can use them for protein synthesis. We can feed them into the citric acid cycle within the liver, and of course both our glucose and our proteins, certain amino acids can be used to build triacylglycerols that can be sent in there. So our well-fed state, we're going to see this. Also, our pancreas is going to sense when we have glucose in there, and it's going to trigger the brain to signal the release of insulin to the liver to say, hey, here comes the glucose, do all this stuff, turn on all the energy storage components. Okay, so this is just an overview of kind of what's going on in the well-fed liver. Okay, if we look at the pancreas, we have our specialized cells in here. You've got alpha cells, beta cells, and delta cells. We mainly talk about the beta cells because those are the ones that secrete insulin, but our alpha cells secrete glucagon, and our delta cells secrete somatostatin. All these are in this, again, anatomy. Ooh, if you think biochem, I liked biochem because I could understand it all. And when you get into the anatomy, I'm like, how the hell do you see that? Why is this different? But all these are in these islets of Langerhans in the pancreas in there. And these all play their particular roles. We're going to compare insulin and glucagon a lot. We won't talk about somatostatin too much, but all these are in these specialized cells within the pancreas. Okay, so glucose regulates insulin secretion. You're like, wait a minute, insulin regulates glucose uptake. Well, there's kind of a feedback in here. So glucose can enter these beta cells through the GLUT2 transporter, okay? And then it goes into glycolysis and ATP levels increase. The ATP, that increased concentration of ATP means more of it is going to bind to these ATP-gated potassium channels, okay? And our ATP potassium channels are going to close and depolarize the membrane, and that will trigger the opening of voltage-gated calcium channels. So we see this buildup of potassium from one side and the other. We get that electrical potential that's going to open up the calcium channels to depolarize it, and the influx of calcium is going to stimulate the release of insulin from the secretory granules where they're stored in these beta cells in there. Okay, so it seems like a convoluted passageway, but glucose comes in, we make more ATP, we close off the potassium channels, which opens the calcium channels. Calcium comes in, binds to its receptors or components in there, which triggers the release of insulin. All right, which then says, hey, there's glucose. Do the stuff with the glucose. Okay. Sulfa drugs can target these ATP-gated potassium channels. So we have, this is more information than you need to know. This octamer of SIR1 and KIR62 subunits in there. So sulfonylurea receptors, where the SIR comes from there, and SIR drugs will close this, and so it mimics high levels of ATP in that regards, closes the potassium channel, and then causes the calcium channels to open and stimulates insulin secretion. So in the case of type 2 diabetes, where you have insulin resistance, this is basically a way to release more insulin. Okay, we're not getting enough of a response to the insulin, so we'll release more to try and get a bigger response in there. Okay, because we talk about insulin resistance, it's not insulin immunity, it's just insulin resistance. Okay, and this is just a picture of how it works and kind of where our ATP binds and then where our other things bind in here. Okay, all right. Glucagon can raise your blood pressure. Okay, so glucagon is, again, we have a cyclic AMP mediated thing in here. In the liver, glucagon will activate glycogen phosphorylase, and it inactivates glycogen synthase. So glucagon gets released when we have low blood glucose. It's an early sign of hunger, and it's going to stimulate processes which will release glucose and raise the blood glucose concentration. So in the liver, we'll see that F2B, fructose 2,6 bisphosphatase will be, levels will be lowered, so that will inhibit glycolysis and thus will stimulate gluconeogenesis. It'll inhibit pyruvate kinase, so we're preventing phosphatidylpyruvate from being converted to acetyl-CoA. So we're not going to be making triacylglycerols. We're going to be, burning those triacylglycerols for energy. And then this again also favors gluconeogenesis. And then of course our glucagon is also going to stimulate pepsi-K for gluconeogenesis as well. So multiple ways that glucagon acts to help elevate glucose production and increase blood glucose levels. Okay. Glucagon will also affect adipose tissue. All right. And again, this is through a cyclic AMP cascade. At adipose tissue, it's going to activate triacylglycerol hydrolysis so that we can send fatty acids to the muscles and the heart for energy. It's going to activate hormone-sensitive lipase. And what this is doing is saying, hey, these other organs can use fat. We need the glucose to go to the brain because the brain can't use fatty acids. So we want to save the glucose for the brain. As for the other organs, let them eat fat. Marie Antoinette thing there too. Okay. So that's what we see. So if we kind of give an overview of the fasting glucogenic liver as opposed to the well-fed liver, the well-fed, what was the terminology there? Back up there. Lipogenic liver, where we're making fatty acids. Our glucogenic liver, we're going to be making glucose. Okay, so our glucagon now comes in, again, released from the pancreas, says, hey, we don't have enough glucose in here. So we will take proteins that get broken down in the liver to make pyruvate. We'll stimulate gluconeogenesis so they can release glucose to go to the brain. We can also see ketone bodies. This can also come from fatty acids as well, too, coming from the adipose tissue. And our ketone bodies can go to the brain. And of course, our fatty acids can get released to other organs as well. Right. So it's kind of an inverse series of processes. Our fasting glucogenic liver is producing glucose for the brain. Our well-fed lipogenic liver is storing energy for later as lipids primarily, glycogen as well. But, okay. So if we compare, effects of insulin on blood glucose, we can see like the metabolic effect as we release insulin, we're going to see increased glucose uptake in pretty much across all organs. We're going to see increased glycogen synthesis in the liver and the muscle, and decreased glycogen breakdown in the liver and the muscle. We'll see increased glycolysis, increased fatty acid synthesis, increased triacylglycerol synthesis. And the way this functions is by targeting certain enzymes. So we'll look at the GLUT4 transporter. We don't go into a lot of detail, but we've got multiple glucose transporters. GLUT2 is the one that kind of just, in or out, doesn't matter where it's more on. GLUT4 is, hey, come on in, our glucokinase, in the liver, hexokinase 4, we're going to see that activated, glycogen synthase activated, glycogen phosphorylase deactivated, PFK1 indirectly activated, so on and so forth. Through all these. All right. And basically for the glucagon is going to have the reverse effect of all this. They won't necessarily be the same enzymes, but we'll see the opposite metabolic effect there. Okay. We should compare epinephrine and glucagon. Why? Well, because they sort of, they use the same pathways in the fat and the liver. But epinephrine is more like our immediate burst. Glucagon is like, hey, we're getting a little low. It's it's like when my wife texts me, like, hey, can you stop and pick up eggs and bread by the store? And if I forget to do it the next morning, would be epinephrine. It's like, we need eggs now, we need bread now. So epinephrine is the faster, more urgent necessity in here. So just like glucagon, epinephrine is going to stimulate secretion So epinephrine is going to stimulate glucagon secretion itself and inhibit insulin secretion, but epinephrine is also going to break down muscle glycogen as well for that burst of energy. And it's also going to stimulate glycolysis and then lactic acid formation as well. So it's going to increase this fructose 2,6 bisphosphatase to stimulate PFK1 to go through, our first committed step of glycolysis. Okay. All right. So what happens after you eat a meal over the next 4 hours? If you notice, we'll say maybe you have breakfast at 7 o'clock and you have lunch at maybe like noon and you have dinner at say 5. So that's roughly like 5 hours in between those meals, right? Maybe you snack at 9 before you go to bed. Okay. So every 4 or 5 hours we're eating. Fuel use, immediately after a meal, we're going to use glucose as our primary fuel because it's so abundant. Insulin is going to stimulate glycolysis and glycogen synthesis. After a couple hours of our meal, postprandial, our blood glucose levels are going to start to drop. We're going to tail off there, and then we can start using glycogen. The liver glycogen will start releasing glucose to maintain those levels. After 4 hours, the same process is occurring. You're going to see glucagon is released because like, hey, we're getting a little low on energy here. We're also going to see an increase in triacylglycerol hydrolysis, and fatty acids become an increasing proportion of the fuel that is used for the muscle and the liver. Okay. Come on. There we go. If we fast for a long time and don't bring any new food in, then we begin to see some other effects in here. One of the things we'll start to see is that muscle tissue will be used for fuel. If you think of the pictures that you've probably seen in your history books of like concentration camp prisoners, or if you go countries that have endured extreme famine, you get these very, very skinny people that have almost no muscle mass as well as no fat. It's because the liver is deaminating or transaminating amino acids and converting the amino groups to urea, but they're using those carbon skeletons of the gluconeogenic ones to make glucose and providing glucose for the brain in there. The fatty acids get oxidized to acetyl-CoA, but our oxaloacetate is very low, so we end up making ketone bodies in there, and that gets exported to other tissues. But what happens is we can see these ketone bodies will lower the blood pH. So we'll see ketoacidosis. The blood becomes more acidic, and over time that can result in coma and death. Okay. So for prolonged fasting or type 1 diabetes, our amino acids, see our urea exported to the kidneys and excreted as urine. Citric acid cycle intermediates will feed in there as we can and try and make oxaloacetate. We'll try and produce glucose as we can and get that to the blood, to the brain. Our fatty acids go to the liver and will get broken down. We'll make our acetyl-CoA. Our acetyl-CoA will favor this ketone body production. Ketone bodies can go to the brain, and of course, excess ketone bodies can end up in the urine as well. You can also, if you make acetone, this is why somebody with really low blood sugar who's diabetic, they used to check the risk of their breath smells like acetone. And it's like, oh no, they're very hypoglycemic at that point. Right? If we look at long term, so here we are at days of starvation, you can see after 1 day, beta hydroxybutyrate levels shoot way up. We're making these ketone bodies. So if you don't eat for a day, you're going to see a massive increase in ketone bodies, specifically beta hydroxybutyrate. Your glucose, you see it drops, but when we get to 40, it's trying, your body is going to try and maintain those blood glucose levels, burn glycogen, convert proteins into glucose. It takes, I think I said it was under 40 is where, you start to get into the, warning, the danger signs in there. Under 30 is when it gets really bad. And you can see it takes about a month for you to hit a 30 millimolar concentration of glucose, which is where you're like, yeah. You also see free fatty acids being used more often, but it's not as dramatic a shift in here. We motivate our adipocytes to release free fatty acids for fuel, but maybe it doubles over like a couple weeks time. And that's a weird thing. Even in starvation, 75% of the cleaved free fatty acids are converted back into triacylglycerides in the adipocyte. Now, gradually you start, having fewer and fewer fat acids there, but it still cycles back in there because your adipocytes want to store for later. And we can see over time, our acetoacetate and our acetone increases as level 2. Okay. All right, I think we can cover just a little bit more. Cortisol is our stress hormone. This acts on liver, fat, and muscle. And, in short term, this is a response. It promotes fatty acid and glucose release in response to pain, anxiety, fear, infection, low blood glucose, whatever your body needs energy to deal with whatever's causing, whatever environmental factors are causing this. So it can change the types and the levels of the enzymes rather than activating existing ones. So in fat, it's going to increase fatty acid and glycerol release. And liver, it's going to stimulate the synthesis of Pepsi K for gluconeogenesis. And the muscle, it's going to break down protein to export in there. Cortisol basically counteracts the effects of insulin. Okay, so if you're constantly stressed or constantly in pain, you can lead to this. You can get what's known as adrenal fatigue. You basically, start becoming inured to cortisol. You adapt to it, to the high levels of concentration in there, and that can ultimately lead to cortisol release being compromised, where it doesn't work the way it's supposed to. And then the effects of this, this constant high levels of cortisol, and then the inability to release it as needed, leads to damaged bone and muscle structures and can affect the immune system. So this may be one of the reasons why, if you're under constant stress, you're prone to getting sick, or if you're constantly living in fear, an unhealthy environment can affect your physical health. What's that? Yeah.
Like, if you don't, what if you don't know, like, the difference between stress and not stress? As a person that grew up kind of like very shy, and also like also kind of like microaggressions and stuff like that. After time, when people be like, "Oh, you're probably stressed. You're stressed," I'd be like, "I feel normal." Like,
Yes.
but I'm
So,
like, is that because I'm always there, or like, because I know people who are the same. They're like, "Oh, I'm not stressed. That's just everything."
and this becomes a question between our consciousness, our ability to think, and all we're talking about right now is animal stuff. Animals go through all this as well too. You can stress out animals and see them get unhealthy and die. So how, we have the capacity to understand what's going on and then hopefully identify it. It's not always easy. It's difficult. The story I like to share for this constant cortisol effect. I know someone whose grandmother was married to, old times when things were different. She was in a very abusive marriage. This is religiously is like, divorce is not an option. You can't, you just got to stay there and you just got to stay there and deal with it. But it was so bad that she started, squirreling away money to leave. And over the course of a couple years, had accumulated a couple $1,000s, several thousand, maybe enough that she was about ready to actually leave. And then her husband found it and of course took all the money because, he was the head of the family. It was his, she had no rights anyway, something like that. And then she died within a month just because of the stress from that. So, yep, that can be the stress of losing someone that you depended on so much is going to see this cortisol effects in there too.
It was like a lady, her and her husband been married since like 18, they were like 80.
Yeah.
And I think he had passed away like a couple years before she did. She eventually died of a broken heart, but she was so used to being around him her life. So
if you concentrate, you can lower or raise your heartbeat, right? If you think, I want my heart to beat faster, you can actually, so you have this, was it psychometabolism? You have an ability to kind of raise or lower various effects in there. We can do this consciously. We can train ourselves. I used to do this. Whenever I'd go to the doctor to get my blood pressure taken, I was like, how low can I get my blood pressure? And I just sit there and, at one point it was like 98/49. I'm like, what in the world? It's like low blood pressure in there. So And you
How
can
do you
kind
do
of
that? I'm trying.
so. The best thing is to practice, and then I used to do it. Like you remember when like all the pharmacies used to have a little seat in there, and you put your arm in and then read your
They
blood.
still got those.
Yeah. Yeah, okay. Good. Yeah. Yep. So you can go find one of those and play around when it's not busy, and just say, all right, let's see what I can do with my blood pressure. And just kind of every 5 or 10 minutes, and just kind of say, I want it to be low. I want to calm low blood. But they're I'm a terrible way of explaining it because I just kind of played around with it. But you can probably find things on Google. Just Google up psychometabolic. Yeah. And yeah, you see things like you've ever been to the hospital for like emergency or something like that. You might notice, oh, boom, there's a big spike in blood pressure because you're so stressed out. Because of course, cortisol is gonna do all that. And then if you can kind of, it becomes, how do we reconcile our conscious mind with our animal body? And it's not always easy. There's a lot of stuff that, there's a lot of stuff that goes on behind the curtain that we don't see. How does our body work? As we learn more and more, we can understand more about it. But even then, there are things that, well, I can't wish my cancer away, or whatever. But but it's interesting to see how the active mind can influence these metabolic effects in here. Okay. Not going to get to it, but we're going to talk about diabetes next week. And this was the picture I wanted to get to here. Our leptin mouse. Look at that big puffy fat mouse on the left there. Yeah. Yep. It is a leptin deficient mouse, versus one that's treated with leptin. And unfortunately, these slides changed. The original version, they used to have, the pharmaceutical companies thought they were going to make billions off of this. Give a pill and people lose weight, but it turns out it's more complex than that. Yeah, yeah, yeah. So yeah, we'll talk about leptin and ghrelin and diabetes on next Thursday. So we won't have class on Tuesday because it's research day. This is what I'm trying to get to. But yeah, okay. And but we will probably hang out. And I think there's gonna be like a chemistry mixer at like 9 to 11 down there. I've not seen it yet, but I will see them all. I will go and do cool stuff on research day.